In our lab, we observed the growth cycle of yeast through measuring their population growth over time. Here is how we achieved this. First, we measured out 0.2 grams of yeast into a weigh boat on a scale. We then measured out 100 milliliters of distilled water into a graduated cylinder. We then mixed the 100 milliliters of distilled water and 0.2 grams of yeast in a 100 milliliter glass container and stirred until it was a one uniform mixture. Next, we measured out 13 milliliters of grape juice into a graduated cylinder. After we were done measuring the grape juice, we measured 37 milliliters of distilled water so that when added to the grape juice, the total volume of the solution would be 50 milliliters. We then mixed the two liquids into a smaller 50 milliliter glass container. This solution will act as the yeast cell's home. Now that we have our two solutions, our yeast culture and our grape juice / distilled water mixture, we took half of a milliliter from our yeast culture and added it to our grape juice / distilled water mixture, which, as I mentioned before, will be the home for our yeast for the next few days. We then labeled our sample with the date and our names. The date so we could tell how old it was, and our names so that we did not confuse our yeast samples with the other classes. Also in our investigation, we found that it was very hard to write on curved glass bottles. We then put our culture in the incubator for a few days. The amount of days spent in the incubator varied depending on the sample. We then proceeded to clean up everything. We did this by rinsing everything out and putting it on the drying rack. We rinsed out glass containers, graduated cylinders, and pipettes. We did this every day during the week for 5 days. 7 days after we had prepared our first culture, we created slides by stirring them and then taking a drop of the solution and putting it on a slide. We then observed these slides under a microscope and counted how many yeast cells we saw. Here is an example of what we saw on day 6. There were 10 times as many cells seen because I diluted it, reducing the yeast concentration, making it 1/10 of what it originally was. Here is the data we collected over the course of the lab. Here is a graph of our data showing the population fluctuations of our yeast cultures. Data and observation questions. 1. Infer. Why was grape juice used to prepare the yeast cultures instead of just plain water? Grape juice was used instead of water alone to provide nutrients for the growing yeast population. 2. Why will you locate the yeast cells under low power, but switch to high power to count the cells? We located the yeast cells under low power, but switched to high power to count the cells because searching for yeast cells with the high power objective would be hard because of the low field of view. 3. Suppose you have to do one dilution of your culture before you are able to count the yeast cells. If you count 21 yeast cells in the diluted sample, how many yeast cells were in the same area of the undiluted sample? The answer to this is 210 yeast cells. 4. What do you think will happen to the yeast population between day 3 and day 7? Give a reason for your answer. Day 7 will have the largest population because the yeast have had the most time to multiply. Day 3 will have the least amount of yeast because the yeast have had the least amount of time to multiply. Critical thinking and analysis questions. How does your data compare to this graph? Is it the same or is it different? Our data is different from this graph, as our data reaches its peak but then declines. What density-dependent limiting factor might cause the yeast population to decline rather than level off? The amount of resources would be a limiting factor, because when yeast are competing for resources, some would not get enough resources and starve and die, causing the yeast population to go down. 3. Describe how you can modify the procedure to ensure that the results were not affected by any variables other than age. A way that we could achieve this is by giving the yeast unlimited access to grape juice and an unlimited amount of space. You do not notice a yeast cell near the edge of the field of view. What effect would the dilution factor have on the size of the error? If you diluted it once, it would miss 10 cells. Twice, it would be 100, and so on. 5. For the cultures you observed in this lab, your teacher used a culture medium that was 25% grape juice by volume. How would changing the concentration of the grape juice affect the rate of growth? Form a hypothesis and design an experiment to test your hypothesis. The concentration of grape juice would probably increase the rate of growth. This could be tested by doing a separate set of glass containers with yeast, but this time the amount of grape juice will be 50%. You would then compare your results with the first set. Conclusion. The purpose of this lab was to observe the growth cycle of yeast through measuring the population growth over time. We achieved this by growing yeast in small containers and measuring their populations under a microscope. We found that there would be occasional large drops in their populations that may be due to limiting factors such as little resources or space. But some drops are very extreme. One time, population decreased from 380 to 5 in one day. To further our knowledge, we could redo the experiment and see if the large drops are still present.